Okay, hello. Welcome to today's lecture on lipid metabolism and regulation. So I'm Associate Professor Dr. Muhammad Razif Shahriar, and I will go through this lecture with you, which will explore the digestion, absorption, transport, metabolism, and also regulation, with a focus on how these processes are crucial for energy regulation and management. So these are the learning outcomes. So by end of this lecture, you should be able to explain the integrated metabolism um, of lipid during covering the process in adipose tissue, the liver, and beyond. And number two is you should be able to understand and explain how the regulation of lipid metabolism in our body. So as an introduction, lipids are groups of molecules that includes triglycerides, phospholipids, sterols, and despite their bad reputation that we always hear, um, these lipids are vital for storing energy, building cell membranes, and acting as chemical messengers. So think of lipid as the body's fuel tank, where the triglyceride acts for energy storage, and also it is also a construction bricks where the phospholipids forms the cell membranes, and it also acts as the blueprint and messengers via the sterols and hormone productions. So their hydrophobic nature makes them perfect for the as to act as a energy storage, but they require special transport mechanism. So that will be discussed after this. So what happens to lipid after entering the human body? So once you eat fats, they undergo a journey of digestion, absorption, and transport. So in digestion, the lipids are broken down into simpler components like glycerols and fatty acids. And during absorption, these components are being absorbed into the intestinal cells. And it will be transported as a bake, where it's packaged with the lipoprotein for distribution. So why do you think fats needs to be broken down into smaller components before absorption? So actually, the breakdown ensures the molecules are small enough as protein and carbohydrate and so on to cross the intestinal lining and enter the bloodstream. So this figure here shows the triglyceride digestion in GI tract. So triglycerides are the most common dietary fats, and their digestion starts in the stomachs and finishes in the small intestine. Okay, so if you look at this figure here, so if you look at this figure here, so in the mouth and salivary glands, some hard fats are being melted as they reach body temperature, and the sublingual salivary gland in the base of the tongue secretes the lingual lipase. And then fat enters the stomach, and in this stomach, the stable lingual lipase initiates lipid digestion by hydrolyzing one bond of triglycerides to produce diglycerides and fatty acids. So the degree of hydrolysis by lingual lipase is slight for most fat, but may be appreciable for milk fats. Okay? So the stomach's churning action mixes the fat with water and acid, and a gastric lipase accesses and hydrolyzes only a very small amount of fat. So after that, it moves to the small intestine. So the bile will flow from the gallbladder via the common bile duct. So this bile will act as an emulsifier. So, so the fat will be emulsified with the help of bile. And the pancreatic lipase flows from the pancreas via the pancreatic duct. And this pancreatic lipase will help to break down the emulsified fat, which is the triglycerides, into monoglycerides, glycerol, and fatty acids before it is being absorbed. So, and next, in the large intestine, some fat and also some cholesterol, which are being trapped in fiber, exit through the feces. So, so this figure shows the absorption of lipid, where once the lipids are being broken down, it's being absorbed in the small intestine. Okay? So monoglycerides and also free fatty acids enters the intestinal cells, and inside the cells, they are reassembled into triglycerides and packaged into chylomicrons. Okay? So chylomicrons are the delivery trucks, okay, loaded with dietary fats. So that they will travel through the lymphatic system before entering the bloodstream to deliver the lipid to the tissues. So if you want to follow the diagram here, the large fat droplets, okay, enters the small intestine after the meal. So the bile just now mostly, and also lecithin will emulsify the fats into smaller particles. So the bile is from the gallbladder duct. So the lipase breaks down the fat, pancreatic lipase breaks down the fat into fatty acids and monoglycerides. So the fatty acids are the yellow ones, and the monoglycerides are the green ones. So the monoglycerides and fatty acids are being absorbed as cells through the brush border and then reformed into triglycerides. So this is already inside the cells of the intestine linings. The short and medium chain fatty acids enters directly into the bloodstream through the cardiovascular system, while the triglycerides are being combined with cholesterol, proteins, and other phospholipids to form chylomicrons, and this will enter the lymphatic system. So we have the cardiovascular system and also the lymphatic system. So moving on to the transportation of lipids. So lipids needs transporters because they don't dissolve in blood. So blood is mostly water, right? You remember? So they don't dissolve in the blood, water. So lipoprotein acts like the taxis. So they carry lipids to the tissues throughout the body. So lipoprotein is a class of lipids and protein, and they are used as a transport vehicles for fats in the lymph and also in the blood. So there are 4 types of lipoproteins, from the largest to the smallest. The first one is the chylomicrons, the largest one. And then we have very low density lipoprotein, or VLDL. So chylomicron just now transports the dietary triglycerides, and VLDL delivers the triglycerides synthesized by the liver. And then we have LDL, low density lipoprotein. So LDL delivers the cholesterol to tissues, which can lead to the plaque build up if the cholesterol are excessive. And finally, we have HDL, high density lipoprotein. So high density lipoprotein collects excess cholesterol from all over the body for disposal. So you have heard about the bad cholesterol and the good cholesterol and so on. So HDL cholesterol is the good cholesterol. LDL, VLDL are the bad cholesterol. So you can think of HDL as the recycling truck. So it cleans up the excess cholesterol in the body, while LDL is the delivery truck, which can clog roads or the arteries if it is overloaded. So each type of this lipoprotein contains different kinds and amounts of proteins and lipids. So the more lipids, the lower the density, and the more proteins, the higher the density. So the next slide here shows the comparison of lipoproteins. So we have known that the lipoprotein vary in their size and also density based on their name. So the more lipids they carry, the lower the density, example like chylomicrons. And the more proteins, the higher the density of the HDL. So the primary component of HDL is protein, and the primary component of chylomicrons are triglycerides. And the primary component of LDL is cholesterol. So the key role of chylomicron is they carry dietary fat from the small intestine to the cells. VLDL carries lipids both taken up and made by the liver to the cells, and LDL carries cholesterol made by the liver and from other sources to the cells, and HDL helps remove cholesterol cells and in turn excretion of cholesterol from the body. So this is another figure showing you the lipoprotein interactions. So the chylomicrons carry the absorbed fat to the body cells, and VLDL carries fat taken up from the bloodstream by the liver, as well as any fat made by the liver to the body cells. And LDL here arises from VLDL and carries mostly cholesterol to the cells. And finally, HDL arises from the body cells, mostly in the liver and intestine, as well as from particles that part of other lipoproteins. So HDL carries the cholesterol from the cells to other lipoprotein and to the liver for excretion. So this figure here shows the lipid metabolism in the adipose cell following a meal. So after a meal, excess energy is being stored as fat in the adipose tissue. And this process are driven by the insulin. So insulin actually stimulates adipose tissue to take up glucose and also fatty acids. And glucose is then being converted into glycerol, while fatty acids are stored as triglycerides. So you can imagine that adipose tissue as your pantry. Okay? So after shopping or eating, you store excess groceries, the one that you wanted to keep as fats for future use. Okay, so you keep it in the pantry. So this pantry is the adipose tissue. So this diagram here shows how triglycerides are being formed in adipose tissue post-meal and highlighting the roles of glucose and free fatty acids in the process. So glucose is being metabolized to make acetyl-CoA, which can be converted to fatty acids, and lipoprotein lipase acts and triglycerides in chylomicron, causing free fatty acids and acyl glycerol to enter the adipose sites. And next, lipoprotein lipase acts on VLDL so that free fatty acids and monoglycerides enter, or monoglycerides enter the cells. The pathways favor energy storage as triglycerides, and this insulin stimulates the lipogenesis by promoting entry of glucose into the cells and by inhibiting the hormone sensitive lipase that hydrolyzes the stored triglycerides to free fatty acids and glycerol. So this slide shows the metabolism in the liver following a fatty meals. Okay? So the liver acts as the body's processing factory after a fatty meal. Okay? So if you take a high-fat diet, so the liver acts as a processing factory. So the dietary lipids arrive as chylomicron remnants and are being processed into VLDL for distribution. So after excess is stored as triglycerides, converted into cholesterol. And during fasting, the liver breaks down the stored fat to produce ketone bodies for energy. So you think of the liver as a manager organizing resources. So they distribute, store, and recycle fat as needed. So this figure here shows how chylomicrons remnants are being taken up by the liver, processed, and redistributed. And you will notice how albumin transports free fatty acids released from the triglycerides. So the first step is dietary nutrients enter the liver through the portal vein, and glucose will be converted into glycogen or glycolysis, or enter glycolysis in the hepatocytes. Amino acids enters the amino acid pool, and some are metabolized to produce pyruvate and oxaloacetate, and serum fatty acids bound to albumin here enters the fatty pools and forms the triglycerides. The chylomicron remnants enters the hepatocytes in the liver by endocytosis and are being taken up by the lysosomes. So the free fatty acids, monoglycerides, and cholesterols are being released in the hepatocytes, and the lipids are reformed to form triglycerides and also cholesterol esters and are being packaged. The triglycerides, cholesterol, and phospholipids are being packaged with apolipoproteins and enter the circulation as the VLDL. So from the hepatocytes, from the liver to the systemic circulation as VLDL. And this VLDL will deliver the triglycerides to muscles, adipose tissue throughout the body. So this slide will discuss about the integrated lipid metabolism in the tissues. So lipid metabolism is balancing act between energy storage and also energy released. So when there is times of energy needs, example during the exercise session or during a low calorie diet, the stored triglycerides will be mobilized by the lipase catalyzed hydrolysis and being released into the circulation as free fatty acids. So free fatty acids will bind to albumin to transport to the cells. And at the cells, the free fatty acids are being oxidized via the tricarboxylic acid cycle for ATP production. So when energy is abundant, especially during post-meal, the excess monosaccharides and amino acids can be converted to fatty acids for storage in the adipose tissue. And when there is insufficient dietary energy, fatty acids can be converted to ketone bodies to maintain functions of brain tissue and also red blood cells. So in short, during post-meal, excess energy is being stored as triglycerides in the adipose tissue. And during an exercise or fasting or in low-calorie diet, the stored fats are being broken down into free fatty acids and transported to tissues for ATP production. So this slide shows the catabolism of triglycerides and fatty acids. So the breakdown of triglycerides or lipolysis is driven by hormones like epinephrines, and triglycerides are being hydrolyzed into glycerol and also free fatty acids. And these glycerols will then enter the glycolytic pathway or gluconeogenesis, and while free fatty acids are being oxidized for energy. So why do you think what do you think triggers this catabolism process? So it might be fasting, low calorie diet, low energy diet, or even exercise. Okay? So let's move on to beta oxidation of fatty acids. So the breakdown of fatty acids in beta oxidation of fatty acid is the breakdown of fatty acids into smaller chain fatty acids and acetyl-CoA. And this will yield energy in the mitochondria. And we know that 1 gram of fat catabolism will produce 9 kilocalories or 37.7 kilojoules. But then beta oxidation of fatty acids is not very efficient because it requires transport into the mitochondria by carnitine through the carnitine acyltransferase system. And it's also less efficiently used for energy production compared to carbohydrate. And they are more preferred to be stored in the adipose tissue. So a single fatty acids like palmitate, C16, produces 106 ATPs, making it highly efficient as the energy source. So when the fatty acids enter the cells, coenzyme A activates the fatty acids to acetyl-CoA with the help from cystoplasmic fatty acid acyl-CoA synthesis, and this uses 2 ATPs. And mitochondria transfers acyl-CoA via the carnitine shuttle. So we have carnitine acyltransferase 1, CAT1, which is in the cytoplasmic site, and carnitine acyltransferase 2 or CAT2 in the inner membrane site. So this is what I told you just now about the cysteine carbon palmitic acid molecule. It will produce up to 106 ATPs throughout the beta oxidation process. Formation of ketone bodies. So during fasting or low carbohydrate diet, excess fatty acids are being converted into ketone bodies in the liver. And this will serve as an alternative fuel for the brain and also the muscles. So the concentration of ketone bodies in the blood is very low in a normal condition. And the concentration will increase in situations of accelerated fatty acid oxidation during body fat reduction, where consume low energy and low carbohydrate diets, and also in uncontrolled type 1 diabetes. So adipose tissues, adipocytes, adipose cells release free fatty acids into the circulation, and this exceeds the ability of tissues to oxidize them. The glucose requiring tissues like the brain and red blood cells cannot use fatty acids for energy, and their need for alternative fuels increases. So the liver can handle excess free fatty acids by converting them to ketone bodies in a process called ketogenesis. So following the beta oxidation, the liver converts the excess acetyl-CoA to acetoacetate, B-hydroxybutyrate, and acetone. So acetoacetate and B-hydroxybutyrate are not oxidized further in the liver, but are being transported by the blood to the peripheral tissues, where they can be converted back to the acetyl-CoA and oxidized through the TCA cycle or Krebs cycle. So this acetone arises in the body by spontaneous decarboxylation of acetoacetate. And the reactions in the ketone body formation occurs only in the mitochondria. Ketone body formation is considered an overflow pathway of acetyl-CoA use, which is another way for the liver to distribute fuels to the peripheral cells. So when there is a mild increase in ketone bodies, we call it as ketosis, and these are expected during fat loss and usually pose no harm. But in uncontrolled diabetes mellitus, in starvation, or with prolonged consumption of a very low carbohydrate diet, ketone bodies can rise to dangerous levels that lower the pH of the body, and ketoacidosis happens, or diabetic ketoacidosis happens in the. So moving on to synthesis of the fat. So synthesis of the fatty acids occurs in the cytoplasm and endoplasmic reticulum, the cell, and it is chemically similar to the beta oxidation process, but with some differences. So it starts with 2-acetyl-CoA, where one is converted to malonyl-CoA by carboxylation, catalyzed by the enzyme acetyl-CoA carboxylase, ACC, and the only regulatory enzyme of fatty acid synthesis. Next, both molecules have their CoA portions replaced by a carrier protein known as acyl carrier protein, form acetyl ACP, and malonyl ACP. So joining of the fatty acids ACPs, in this case acetyl ACP with malonyl ACP, splits out the carboxyl that was added and creates the intermediate at the upper right in the figure at left. From this point forward, the chemical reactions resembles those of beta oxidation in a reverse manner. So acetyl-CoA carboxylase, which catalyzes synthesis of malonyl-CoA, is the only regulated enzyme in fatty acid synthesis. Its regulation involves the allosteric control and covalent modification. And the enzyme is known to be phosphorylated by both AMP kinase and protein kinase A. Dephosphorylation is stimulated by phosphatase, activated by insulin binding, and this phosphorylation activities activates the enzyme and favors its assembly into a longer polymer, while phosphorylation reverses the process. Citrate acts as an allosteric activator and may also favor polymerization, and palmitoyl-CoA allosterically inactivates the polymerization. In animals or human, 6 different catalytic activities necessary for remaining catalytic actions to fully make palmitoyl-CoA are contained in a single complex called fatty acid synthesis. So this includes the transacylases for swapping CoA with ACP on acetyl-CoA and malonyl-CoA, synthase to catalyze addition of the 2-carbon unit from the 3-carbon malonyl ACP in the first step of the elongation process, reductase to reduce the ketone, dehydrase to catalyze removal of water, and reductase to reduce trans double bond. So this is the fatty acid synthesis model with positions of polypeptides, 3 catalytic domains and their corresponding reactions. Elongation of fatty acids. So elongation to make fatty acids longer than the 16 carbon occurs in the endoplasmic reticulum and is catalyzed by enzymes described as elongases. So mitochondria can also elongate fatty acids, but their starting materials are generally shorter than the 16 carbon. The mechanisms in both environments are similar to those in the cytoplasm, where a malonyl group is used to add 2 carbons, but OA is attached to the intermediates, not the ACPs. Whereas in cytoplasmic synthesis, employs the fatty acid synthesis complex, the enzymes in these organelles are separable and not part of the complex. Desaturation of fatty acids. Fatty acids are synthesized in the saturated form and desaturation occurs later. Enzymes called desaturases catalyze the formation of cis double bonds in major fatty acids. These enzymes are being found in the endoplasmic reticulum, ER. Animals and humans are limited in the desaturated fatty acids they can make due to an inability to catalyze reactions beyond carbon 9 and 10. So, therefore, humans can make oleic acid, but cannot synthesize linoleic acid or linolenic acid. So, these two must come from the diet. Consequently, they are also called as essential fatty acids because it must be provided in the diet. Let's move on to the regulation of lipid metabolism. So hormonal hormonal regulation done by insulin and glucagon for lipid metabolism. And there are also allosteric controls where the modulators are acetyl-CoA carboxylase, like citrate, palmityl-CoA in fatty acid synthesis. Another modulators are HMG-CoA reductase for cholesterol in cholesterol synthesis, and carnitine acid transferase 1 in malonyl-CoA in fatty acids oxidation. Lipid metabolism are also regulated using molecular or gene at the gene expression level. So this is the allosteric control of fatty acids. Enzymes like acetyl-CoA carboxylase are being regulated as by the allosteric modulators, where citrate activates synthesis when energy is abundant, and palmityl-CoA inhibits synthesis when fat stores are sufficient. This figure here shows the allosteric control of cholesterol synthesis. So the enzyme HMG-CoA reductase is a key player in cholesterol synthesis and is regulated by the feedback inhibition. So high cholesterol levels inhibit the enzyme, reducing the synthesis. And this is the target of the statins, common cholesterol lowering drugs. That's all for today's lecture on lipid metabolism and regulation. Thank you.